Okay. So yeah, I was saying that. So welcome to this course, week 5 content of cell culture technologies. So today we will be talking about the hippocampal neurons culturing and how what kind of media we are using. Some basics of the drug discovery processes of how different methods are used for you know. Clearing the methods are used for uh, giving a green signal for a drug to be used in the clinical setup. So some basic knowledge about the cell culture, the different kinds of media requirement, the different kinds of the pH balances and all those things. Some drawbacks of why 2D cell culture is not a very good option and not very relevant to physiological conditions. So those things we'll go through. So let us begin. So the cell culture in vitro requirements, as we all know and as we are been talking about from long time, that appropriate medium is required for supporting the cell growth. So appropriate medium in the sense that different cells require different composition of various amino acids. Some require greater concentration of some basic amino acids. Some require greater concentration of some acidic amino acids. So depending upon what amino acids are there, what other nutrients in the sense glucose, some require sucrose or whatever carbohydrate sources a particular cell requires for its growth. So depending upon that, there are different composition of medium. We will not go into details of medium today, but later on. But there are the most commonly used medium that we use is DMEM or Dalbacos modified eagle medium. So this generally consists of all the essential amino acids and glucose and all the glucose and other ions, etc. So for maintaining proper osmolarity, for maintaining proper nutrient composition. So appropriate medium in the sense that some cells, like some neuronal cells or some specified cardiac cells, etc., those cells require medium which is not maybe DMEM. So that time also there are like medium like HAMS F12K. So all those medium has so this is HAMS F12K. So here in F12K, K means lysine, so it has a greater concentration of lysine. So this is a positively charged amino acid. So depending upon the requirements of the cell, different kinds of medium can be used. So but most commonly used is DMEM. So DMEM goes with mostly all kinds of cells, and it is like very optimized composition. So appropriate medium supporting the cell growth is very important, of which defined medium means that it does not contain serum. So sometimes so what is serum? Serum means the component of the blood except your red blood cells. So those component of the blood is essential for the growth of for essential for different uh, uh, nutrients, as well as protect, because those serums contain certain antibodies and some certain certain chemicals like immunoglobulins or some chemicals which helps the cells to be in a more physiologically relevant environment. So if this serum is not there, sometimes why we don't use serum is when we are transfecting the cells, because we don't we like so some transfection reagents may have an adverse effect or some reactions with those serum which will not allow the cells to get transfected. So that time we don't use serum. But also some other cells, some cells, they don't require serum for their normal growth. That has been optimized. But generally, the cells that we grow in our lab, we grow it in presence of serum, and it is and also we should not exceed the serum concentration. Or so we use basically fetal bovine serum, that is known as FBS. So fetal bovine serum is from the calf, like from the calf of cows. So this fetal bovine serum should be approximately 10 to 20%, should not exceed more than 20% of your composition of the media. So this needs to be added separately. So this is percentage. So basically, this is not X. 1 second, let me erase it. So 10 to 20% of FBS needs to be added for proper growth, should not exceed more than 20%. So DMEM is the media composition that we generally use, and to that DMEM we need to add 10% or 20% of FBS. For example, if we are using 1000 ml of media, in that 1000 ml we need to use only 100 ml of FBS and 900 ml of DMEM. Okay, to get the proper composition of to get the proper percentage of FBS in the media. So when we are using this kind of composition, we have to we have to inactivate. So there is also one process where we inactivate the FBS. So we inactivate the FBS so that all the proteins that were that might like the proteins which are so basically we are taking it from calf. So if those calf proteins might interact with human cell lines or mammalian cell lines that you're working on, so just to inactivate those proteins we have to inactivate FBS. So the media composition and media requirements and all we will discuss in details in later on classes. So yeah, for now it is important to know that there are different compositions of media depending upon what amino acid, what source of carbohydrate we are using, what other nutrients, additional nutrients is required or not. So apart from that, we also require substrate for cell attachment, that is the ECM. So ECM also depends upon what cell line you're using. There are different kinds of ECM as we discussed in last class: polydilysine, concanavalin, and there is what other so these substrates are there. So depending upon whether we want to plate it in 2D or 3D, like whether we want the model system to resemble more as compared to the physiological condition or more as less or like whether we are willing to spend more of our time into culturing those 3D cells. So it depends upon what kind of substrate we are using and whether we truly require it or not. So 3D cell culture is again another kind of like a little bit different from 2D cell culture. Also, it is different in the sense that the cells which are seeded in a 3D environment, when you are observing it under a microscope, it might be a little difficult to focus, and we might require good characteristics like of the microscope, like Z stacking, to see the proper morphology of the cells. So depending upon what is your requirement, if we want to test a particular chemical, test a particular drug which needs to be more relevant, so that time 3D cell culture becomes important. Otherwise, if some signaling pathways you are interested in, or you are interested in some kind of imaging or some protein purification or something, so for that reason 2D cell culture is enough. But for more pharmaceutical industries, or for more drug related processes, those things which requires a more physiologically relevant environment, for that 3D cell culture is required, and so appropriate substrate be needed. Also so also we need to have proper gaseous exchange or pH balance in the cells, so that the proper pH is maintained. Ideal pH should be between 7.2 to 7.4, that is the buffering pH range. And so this is maintained by the sodium bicarbonate system, sodium bicarbonate buffer system. So we have carbon dioxide in our incubator, and we have So carbon dioxide in our incubator, which helps in maintaining the bicarbonate buffer system. Also in our media, we need to add sodium bicarbonate. So in DMEM, sodium bicarbonate is present, which helps in maintaining the bicarbonate, buffering. So sodium bicarbonate is also present. So there is different composition of media. It is available online. It is available in cell culture handbook. So DMEM has a particular concentration. So those things you can look into. And so the basically appropriate medium, appropriate substrate, proper gaseous exchange, like proper pH balance, and also a defined system, whatever you are using, but whatever you want to use it, we should use it for the entire set of our project. We should not change the system frequently, because that will lead to error in our results. So for example, if you are using a particular cover slip from a particular company and coating it with a particular substrate, we should be using it ideally throughout our experiments, throughout the project. And we should not change the ideally we should not change the company of from where we are getting the cover slips, or from where we are getting the coating substrate, because little bit purity or little bit changes in the composition can give a very big effect on your results. So defined system, and also very important, another very important criteria for cell culture is plating equal number of cells in the dishes or cover slips, so that it is uniform and the experiments are repeatable. So if the cells, for particular, like if any kind of experiments, we need to do it in biological triplicates. So there is a difference between technical triplicates and biological triplicates. So technical triplicates means at one time you have 3 different cover slips where you're doing the experiment. So you are having the result from 3 different cover slips. So that is technical triplicates, like at one time 3 different cover slips. Biological replicates means this 3 different cover slips, like suppose in one day of experiment you have done 3 cover slips of experiment. Second day again you are doing 3 cover slips of experiment. Third day again we are doing 3 cover slips of experiment. So this day 1, day 2, day 3. So this is day 1, this is day 2, and this is day 3. So all these days, so when we are doing experiments on different days using different passage number of cells, I'm sure you remember what is the passage number. So whenever we are trypsinizing the cells from one particular passage, from one particular dish and putting it in the next dish again, so the passage number increases by +1. So every day, if we are doing one set of experiments, so every passage, like every time the passage number of the cells increases. So when the passage number of the cell increases, so here suppose the cells are in passage number 5, here it is in passage number 7, and here it is in passage number 9. So this is when we are saying it as biological triplicates. So here every day you are doing, like different day you are doing experiment, and so this is biological like biologically you have to validate the result, and it should be repeatable. So suppose one day you are doing in P5, so on day 1 when you are using P5 passage number, you are getting one set of results. P7 also should be similar set of results, should not be deviated from the results that you got in P5. So there might be little deviation, that is okay, because cells are heterogeneous population, it is not homogeneous. Like some cells will show some kind of effect, some cell will not show that effect. But as a whole, when we are taking the results from all the 3 cover slips together in this particular day, control versus, so this is suppose your experimental 3 cover slips, and control you should have again 3 cover slips. So all the results from the control versus experimental should be repeatable when you're doing it the next day as well. So for the experiments to be repeatable, that is a very big issue in kind of wet lab experiments, because very minute changes in your cell culture like techniques or cell culture methods might lead to a great change, and that might not be very feasible because you have to repeat it a multiple number of times. So in order to avoid it, we have to continuously keep in mind that whatever cells we are plating in the dishes or cover slips, it should be equal number. That should be like really a very important thing to keep in mind. And also we should not ideally change the cover slips company, or change the different coating material, whatever you're using. Initially you have to standardize that which coating material works, which cover slip works, which dish works. Once it is standardized, that is it, and that you have to follow it through the rest of your experiments. So these things we have to keep in mind when we are culturing our cells, so that we get repeatable results, and biologically that has to be like replicated. It should be replicatable. It should be replicable, and not like we should not be like you're getting totally opposite results when you're replicating it. So that is what is about biological replicates and technical replicates. And so the cell culture methods, again, just briefly I'll go through. I had already discussed about how cells for primary culture and cell lines are being isolated. So here again, suppose there can be two ways. Like there are sources from where cells are isolated. One is like organ, which is like not a tumor, like not tumor. It's just an organ, maybe a brain, heart, liver, kidney, whatever. And there is another source which is a tumor containing organ. So the cells which are isolated from this particular organ, these are going to be transformed, and they will be they will generate cell lines. So these are basically cell lines, and the cells which are isolated from an organ, these are going to be a primary cell culture. So this is not going to divide. So basically this is not dividing cells, and this is primary cell culture. So cells are isolated from these organs using enzymatic or mechanical action. Similar is the case for the tumors, and then they are put into culture vessels where the cells grow. Cells are basically, when we are isolating this organ, so we have to dissect the animal, we have to isolate this organ, and this organ, then this organ has tissues. So these tissues have cells which are very, very much closely attached. So we need that the attachment between the cells has to be broken. So for that we are using mechanical disintegration or enzymatic action, which will break the adhesive forces between the cells. So instead of the cells getting in clumps, the cells will become behave as an isolated particle. So the cells has to be isolated. So here again we can see that the cells are isolated, and they will have different characteristics. Now each of these cells, although they are isolated from the same tissue, but each of these cells behave differently. In the sense, what I want to say is that all cells might not be as healthy as the other. All cells might not be from the same part of the organ. For example, this organ is really big, so some cells might be from this area of the organ, some cells might be from this area of the organ. So depending upon which area you are taking on, the cells might show changes in their behavior. So whenever we are doing an experiment, we generally consider whatever cells are there in your field. So behavior wise, if it is different, it should not be very, very different, because it is from the same organ. So always we are averaging, so we are taking the average of whatever results we are getting. So whenever we are carrying out an experiment, so each of these cells, whatever behavior it is showing, so each of these cells behavior is individually analyzed, and then we are clumping it together into an average value. So that is what is control versus experimental value. So once we are getting the isolated cells, and then we are allowing it to grow, if it can divide, so it will become confluent. So again, confluency is the term where we confluency is the term where how much cells are basically covering the surface area of the cultured vessel. So depending upon that, we have 40% confluency, 50% confluency. So how much area the cells are covering? So if it is covering 50% area of the entire surface, then it is 50% confluent. So once the confluency is reached, then we have to subculture it. If the cells are not dividing, we are not going to get any particular confluency. We will it's not going to divide. So whatever confluency we are having, we have to work with that. And so for primary cells, we have to make sure that whatever cells are here, suppose in your culture vessel, if we are plating it for experiments, so if we are plating, suppose 4 plates for experiments, we have to plate equal number of cells in each of the 4 plates. So depending upon how many cells are there initially after mechanical disintegration, we have to count the number of cells in hemocytometer and then equally plate it. Or else another method that we can do is we can we can centrifuge the cells and the pellet. Suppose in pellet we can add 400 microliter of media and add 100 microliter. So it is again equally split. So in that condition, we have to carry out our experiments. In case of cells which are isolated from tumors, so they are, they can be subcultured and they can be grown for a numerous number of times, and cell division can continue up to indefinite divisions. So here again we can, these are transformed cells basically. So these are going to be, this can go on for a long period of time. But for primary cell culture, we have to be very cautious, because these cells, they don't divide, and we have to carry out our experiment within that stipulated period of time. Otherwise it will die, and then again we have to repeat the process. So drug discovery process relies on this in vitro cell culture technology, where we are using cells of relevant organ for against which the drug needs to be tested. So it relies on this in vitro cell culture model before animal trials, because it needs to be validated that it is working in vitro models. Then only animal violence or animal trials is allowed. So after animal trials, then goes to human trials. So first step for any drug screening process is in vitro cell culture. And in vitro cell culture model, so whatever cells, if we want to screen drugs appropriate for neuronal cells, we have to isolate neuronal cells and then screen, and then see what is the drug concentration or what is the toxicity of the drug in that cell line. So depending upon, like, mostly if we are using neuronal cells, then we have to isolate primary cell, the disorders which are affecting the differentiated neurons, then we have to isolate and culture those differentiated neurons and test them. If it is if the drugs are against some tumors, or if it is against some cancer cell line, then it is fine to test it with the cancer, the continuous cancer cell lines. But yeah, so depending upon what requirement we have, we can screen the drugs on this particular appropriate cell lines, and it has to be physiologically relevant, because we need to because, for example, for drugs we have to, this will ultimately, the main goal is to use these drugs in human setup. And if this human setups, if we are not properly screening them in the in vitro level, then it will be difficult difficult to use it in more physiologically relevant conditions. So if it is like, basically, if we are finding some suitable drug for neurodegenerative disorder, if we are testing it in some kidney cell line and then seeing that it is having effect, but actually that is not a very ideal situation, because when we will go to animal trials, that is going to be definitely blocked. And ideally we should not use cells which is not relevant to what disease we are interested in. So so we have to rely on appropriate cell lines for carrying out this drug discovery process. Also, the cell culture models, which are tested after isolating cells from any particular organ or tumor. So, depending upon whether we are working with some neurodegenerative diseases or some cardiac diseases or some cancer, the cell culture models are tested. So, and also we have to test the efficacy. So, for example, now let's take an example as was taught in the lectures. So, maybe if we are testing the efficacy of drugs against Alzheimer disease, then what are the different requirements? What cells are we supposed to use? How, what medium are we supposed to use? So, for example, if we are testing efficacy of drugs against Alzheimer's disease, then the hippocampal neurons is an ideal cell culture model, because Alzheimer's disease is a neurodegenerative disease which affects the growth of hippocampal neurons. Not growth, but it will cause the hippocampal neurons to die. The reason is yet not known properly. So, Alzheimer's disease, why there is a death of hippocampal neurons, and why so why is this disease onset in the late 70s 60s. So it's a late onset disease and neurodegenerative disease. So for proper representation, we need to use hippocampal neurons for testing the drugs. So isolating hippocampal neurons. So hippocampus is a region in the brain which is present in the lower thalamus side. So it is a kind of structure which is similar to the seahorse. That is why it's named as hippocampus. And these hippocampal neurons are isolated from fetus from the pregnant mother's womb. Basically, rat or mice, which is around 18 to 24. So rat and mice, which has a fetus of around 18 to 24 days, the mother needs to be sacrificed and the fetus needs to be taken out from the mother's body, and we have to carry out dissection to isolate and to take out the hippocampus from the fetus brain. Why not from adult? Why not from? Why we cannot take it from an adult? Mice is because hippocampus during, like, if it is in adult mice, it will be difficult to grow it in the cell culture, because it will be totally differentiated and it might not properly adhere, or it might not divide further. But if we are isolating it from a fetus, then the hippocampus, which is it is still it's not still developing, but it is like in a condition where it can still be cultured in vitro condition, and it can still divide and it can still attach. So for that reason, we have to isolate it from fetus. And once we are isolating it from fetus, the particular organ needs to be dissociated enzymatically or mechanical dissociation, whatever works in the lab. So generally tissues which are softer, so we can go for mechanical dissociation. Also, some cases enzymatic dissociation is preferred for some organs which has a better composition of some adhesive structure, adhesive proteins which is better cleaved with those enzymes. So after isolating the hippocampal neurons, we need to enzymatically or mechanical dissociation of the tissue needs to be carried out. And then we have to use a defined medium and substrate, and we need to plate those suspension of cells. So basically we have a hippocampus which is isolated from the cells, from the organ, and then we have to make it into a cell suspension. So here we have the hippocampus, and then we can either use motile pestle or just pipette it up and down. And then when we are pipetting it up and down, so this is a media, or it can be buffer, whatever suits the requirements. We can use media or buffer. once the mechanical dissociation or enzymatic dissociation takes place, then this entire tissue will be broken down into small fragments, and these fragments will have cells in it. And so this is a cell suspension. So this cell suspension then needs to be plated in a defined system. So this defined system will have cover slip or glass. This has a glass cover slip, or just maybe the petri plate, if what petri dish without the cover slip. So it needs to be coated with the ideal substrate, and so this ideal substrate already needs to be coated beforehand only. This plate containing cover slip, or this plate without the cover slip, here we have to pour the single cell suspensions, and then put it in incubator, CO2 incubator, for allowing them to grow. So this goes into CO2 incubator. Now after some days or 24 hours to 48 hours, we can look into the microscope and check how the cells have grown and how the cells have attached. So generally the cells should attach by the time of 8 to 12 hours, but it might not come into morphology. So for coming into morphology, it takes about 24 hours. And for different neuronal cells, we need to also check whether the morphology is correct and whether the cells look healthy or not. So after isolating the hippocampal neurons, we have to go for mechanical dissociation of the tissue, and then we have to use a defined medium or substrate and plate equal number of cells in the dish or cover slip, and then grow the cells under optimized growth condition. So once this optimized growth condition, in the sense that how many hours later we have to give the optimized growth conditions here refers to how many hours later we have to give media change, or how many hours later we have to add some additional nutrients or additional drugs, etc. So these are the optimized growth conditions that when we start working with a particular cell or particular organ, it can you will get used to that duration of time when it needs to be splitted into a different dish. So we have to be very careful when we are using hippocampal neurons, or any neurons for that sake, because these are not dividing cells and they don't have any regeneration capacity. So if the cells are contaminated, or if the cells are lost due to some carelessness, then again the process has to be repeated, and it is not a good example, because again if you have to isolate it, again you have to sacrifice the mice, sacrifice the fetus, and that is not very advisable. So we have to be very careful when we are working with primary cell lines, especially the neuronal cell lines, which tend to get it might be, if it is contaminated, then any kind of cells, if it is contaminated or if it is not healthy, it has to be discarded. So we need to avoid that. So once we are culturing the hippocampal neurons, once we have isolated the hippocampal neurons, plated in our plates with defined medium, defined substrate, and we have optimized all the growth conditions, we know that this condition is suitable for its growth. So now we have the base of the cells against which we are going to do some drug testing for Alzheimer's. So first we have to, for drug testing, first we have to choose the appropriate cell line. Second, we have to see how the cell line can be, cultured, whether it has to be dissected and grown from a primary, whether it has to be a primary cell line or it is a secondary cell line. Once our cell line is optimized, the growth conditions are optimized, next step is to do the drug testing. So first step for this culturing the hippocampal neurons is the dissociating the cells. Like after isolating the tissue or isolating the organ, we have to dissociate it into single suspension of cells, single cell suspension. So there could be two types of dissociation. One is mechanical dissociation and one is enzymatic dissociation. Mechanical dissociation, as I said, we can use mortar pestle, like the thing which has this kind of marble structure for dissociating. So we can use those kind of structure which we have to grind it, like softly grind, not like very rigorous grinding. So soft grinding of this tissue, or just we can pipe it up and down if it is not very hard tissue. If it is very fragile, then we can just pipe it up and down, and that will apply shear force, and it will cause the dissociation of the clumped tissue or the organ. So that is one mechanical dissociation. Enzymatic dissociation are using where we use enzymes that disintegrates the cell-cell adhesion and disintegrates the bonds between the tissues, like the disulfide bonds or the electrostatic interaction. So we are generally using papain or collagenase to dissociate the tissues into single cell suspension. So papain and collagenase, this helps in breaking down the disulfide bond and the collagen respectively. So collagen is again an ECM, the extracellular matrix which is present and which adheres the cell together and helps in the cell clumping. So if we are using collagenase, the collagen will be broken down and the cells which were clumped will become isolated. So cells which were clumped can be, so they are clumped and they have collagen between them for their adhesion. So if this collagen is broken, or if the disulfide bonds between the cells are broken, then this basically each of the cells will become separate entity. So we need to have a single cell suspension, and we can either obtain it using mechanical dissociation or we can obtain it using enzymatic dissociation. So next, the dissociated cells, they need to be grown in a growth medium or buffered solution. So growth medium and buffered solution, as I discussed in the first slide, depending upon what requirements we have. Generally, the medium is used as DMEM medium, but some neuronal cell lines require extra lysine, extra glutamine. So for those reasons, we need to modify the media composition accordingly. Like we need to supplement those extra chemicals or extra nutrients in the media for proper growth. So they need to be coated in appropriate ECM coated. So they need to be, once the cells are dissociated, they have to be plated in appropriate ECM coated plates, which have the composition required for the adhesion of that particular cell line. So poly-D-lysine is a good option for good appropriate ECM matrix for hippocampal neurons. So poly-D-lysine, generally most of the cells are good to go with poly-D-lysine. Only some cells like rods, cones, they prefer concanavalin. Otherwise, poly-D-lysine is a very good ECM substrate for culturing the cells. Next, we need to plate the cells. So for plating, we have to count the cells. So generally, if we want to set up our experiments, so we need to have dishes with equal number of cells. So when we are having, so suppose we need to have 3 dishes where we will carry out experiments. So one, we have control dish. One, we have X drug for treating. Yes.
Can you please explain dissociation of cells once again?
Yes. So dissociation of cells as basically when there is a organ that we are isolating, or an organ that we are dissecting and taking out of an animal, then that organ is like basically a mixed. It has a lot of cells inside it. It is a tissue which contains, which is a combination of lot of cells together. So suppose this is the organ. Suppose this is the liver you have isolated. So the liver has different kinds of tissue in different parts. Basically, the entire liver, suppose this part is having a different tissue, this part is having a different tissue, this part is having a different tissue. Those tissue specific, if you want to specifically isolate a particular tissue, then we need to cut out liver from those region. and then this region's tissue is composed of several cells here. So these cells then they are clumped together because they are they are functioning as a tissue together, like they are functioning as a tissue, and they are in a group and in colony. So this particular tissue, then we need to dissociate it, or dissociation means we have to break the bonds that are present between each of these cells. So each of these cells have a particular bond between them. So this bond needs to be broken down so that each of these cells, instead of this clump behavior that they are showing, so here that they have the clump behavior. So instead of this clump behavior, we need to break all these bonds between the cells. And for breaking the bond, if we are using mechanical forces, then mechanical forces will create some shear action. So as a result of which the cells will get separated from each other, and it will be grown as an isolated like isolated cells and not together, not as clumps. Also enzymatic dissociation we can use, where we are using enzymes that can cleave the proteins that help in cell-cell adhesion. So then also the cell clumping or the cell adhesion will be disrupted, and we will get isolated cells. So basically dissociation of cells means to dissociate the tissues, the tissue containing the number of cells. This number of cells need to be dissociated into isolated variants or isolated cells. So that is what is dissociation of cells. So
Thank you, ma'am.
yeah. So next, once we have the cells, so now we have isolated cells. So we have isolated cells in our dish. So this is suppose our dish, and we have isolated cells over here. Now we want to plate these cells in such a way that in each condition, so this is what I was saying is, we have three experimental conditions, not three experimental conditions, two experimental conditions where we are testing the drug X and testing the drug Y, and we have a control condition where we don't have any drug. So we have to plate the cells in such a way that each of these dish should have equal number of cells. For example, in control also we should have about 500 cells. In X in the plate where we are thinking of giving X drug, it also should have 500 cells, and in the other plate where we are thinking of giving Y drug, they also should have 500 cells. So total in each of these dishes we need to have 500 cells. So this 500 cells, how we can achieve? So we have to basically take the dish from where we want to split into it. So this is the dish where we want to split into three categories. So three other dishes. So this dish, when we are we are trypsinizing the cell, and after trypsinizing the cell, we will get and then we will centrifuge it. So we will get a cell pellet. So this is the cell pellet, and so this cell pellet and one supernatant we will get. So this supernatant we will discard, and we will put fresh supernatant here. So suppose so depending upon how much is your pellet, so you can see so as you are doing the cell culture, you will get used to one kind of idea that this much pellet we have to dissolve in this much amount of media, or how much more media or less media. So depending upon the pellet size, we can dissolve it. So suppose here depending upon this pellet, I am putting around 500 microliter of media, or 100 microliter of media, and from this 500 microliter of media, I am taking 100 microliter of media out, and putting 100 microliter trypan blue. So trypan blue is a dye which is used to stain the dead cells. So we have to count how many number of live cells are there in our cell suspension before plating it. So here when we are plating it, we have to plate it depending upon the number of live cells that are present. So once we get the pellet after trypsinizing the dish, once we get the pellet, we are resuspending the pellet in 500 500 microliter of media. Out of this 500 microliter of media or buffer, whatever, and here 100 microliter we will take out and put 100 microliter of we'll put 100 microliter of trypan blue. So here when we are putting 100 microliter of trypan blue, that means we are diluting the mixture to 1:2, because now the total volume of this mixture is 200 microliter, out of which only 100 microliter is what has the cells. So when we are diluting, so depending upon how much cell and how much trypan blue you are adding, how much dilution you are adding, suppose you are adding 100 microliter cells and 500 microliter trypan blue, then the dilution factor is 1:6, because 100 and like 1:6, yeah. So depending upon how much trypan blue we are adding, the dilution factor changes, and that gets multiplied to the formula of total cell count. So this is the dilution factor. So if I am using 100 microliter of this supernatant and adding 100 microliter of trypan blue, then the dilution factor is half, or half, but dilution, that is the dilution factor is actually 2. Factor is 2 here. So don't get confused between dilution and dilution factor. So dilution means how much diluted you are making the your cell suspension. So if you are diluting it into 1:2 ratio, then the dilution factor becomes 2. If you are diluting into 1:6 ratio, then the dilution factor becomes 6. So the dilution factor is just the reverse of dilution. So dilution means 1:2 dilution, 1:3 dilution, 1:4 dilution, but factor is just opposite, multiplied, just opposite to the dilution, So depending upon how much trypan blue you are adding, so the dilution factor changes. So for here, for our case, suppose I have added 100 microliter trypan blue, 100 microliter cells, then our dilution factor is 2. Next, we have to count the number of cells. So now we will plate this 100 microliter of cells in our this hemocytometer. So this is how a hemocytometer looks like. This hemocytometer looks like an H. There is an H in the center of the plate, and there is we can count the cells over here. So we have to insert our sample in this hemocytometer. So some liquid, so your cell suspension is liquid, and so if we magnify the hemocytometer, like how the hemocytometer looks over here, if we magnify here, we can see there are several grids. So each of these grids have different surface area. So here, as it is mentioned, as this grid has 1 millimeter length, and this also has 1 millimeter. So this square has 1 millimeter. This each side of the square is 1 millimeter. Now, when we are counting the cells, so suppose you have put the cells over here in this hemocytometer grid, and when we are counting the cells, you will see that depending upon what method you are following, mostly people follow method where they will count cells in this grid, this grid, this grid, and this grid. Also, sometimes people follow this square as well. So depending upon how many number of squares you are counting, here in the formula you have to mention, you have to use that number of squares. So if you are counting from 4 squares, you will mention 4. If you are counting from 5 squares, you will mention 5. So for example, if I am counting from 4 squares, I will mention 4 over here. Then we have to count the number of cells in each square. So each square, so we have, suppose for example, so here in this particular square you have cells which are present everywhere in this line. So we should not we should count the cells which are in the top side of the, so this is true for each of the grid. So in each of this grid, if there is a cell which is in the top line, we have to count that cells. If it is towards the right side, that also we can count. In each grid, if the cells is towards the left side, we should not count it. Also, if it is towards the bottom, that also we should not count. So these are some rules which are set like in order to avoid. So these are in order to avoid some errors, and in order to give an optimized way of counting the cells. So this depends upon what lab you are working on. So some lab may be counting the cells which are on the top and right, and not counting the cells on the bottom and left. But some labs may be doing the opposite. Some labs may be doing some different way. But whatever way you are using, we have to make sure that we are using the same way throughout. So once we are, counted the cells in each square, how many cells are there? So suppose total number of cells, we have around 100 cells. So these 100 cells, then we have to multiply it with the dilution factor and multiply it with 10 to the power 4 or 10,000 and divide it by 4 to get the number of cells per ml. So this will give you the number of cells per ml of your suspension. So depending upon how many cells we are getting, we will be able to know that so per ml this much cells are there. So if we want to plate 500 cells, then how much amount of volume or how much volume I need to put in this particular dishes. So that we have to make sure that we are counting the cells and putting equal number of cells in each of these dishes, so that when we are going for our experimental condition, we don't have that biasness that in control cell you had about 500 cells, but in experimental conditions you have only 200 cells. So what this biasness will do is, suppose your control cell, initial starting control cell has 500 cells, and there is no drugs that you have treated in this 500 cells. So all the cells are healthy. But if this drug has some toxic effect, then out of this 500 cells in the experimental conditions, out of the, so if you have different initial number of cells, suppose in control you have 500 and in experimental condition you have 250, then the drug will kill most of the cells. Like if it is killing most of the cells, then the cells number will deteriorate to 50. So then there causes a problem, because we have to normalize it with control, but control already has 500 cells, and that 500 cells were not similar to initial number of cells that you used for the experimental condition. So when we are comparing it with control, so it means that 1 to the power 10 fold decrease has happened. So the drug is very toxic, but ideally it is not. It is just like it has reduced it from 250 to 50, not like from 500 to 550. So we have to be careful that the initial number of cells that we are using has to be similar to control and similar in all the cases, so that it is easier to compare between the different conditions, and that will give you a more statistically relevant result rather than using different number of cells in different conditions. So if we are testing X, Y, and Z drug in cultured hippocampal cell, and we have the evaluation criteria where we have to observe the number of synapses present in the control versus drug treated, then again we have to plate the cells in all these 4 dishes. 1 dish is for control, and the 3 dishes are for different drugs. So this is control, this is X, this is Y, and this is Z. So we have to plate the cells, equal number of cells in each of these dishes. And after plating the equal number of cells, we have to see that, so suppose in control, we are not giving any kind of treatment, no treatment. So this is no treatment, but here we are giving treatment. So now we have to also optimize that how much concentration of the drug we have to use. So how much concentration of X we need to use, how much concentration of Y we need to use, how much concentration of Z we need to use. So this also concentration optimization we need to do, and we need to make a lethal dose curve where we'll get an IC50 value and LC50 value. So IC50 and LC50, I think IC50 means inhibitory concentration that inhibits 50% of the cells, and lethal concentration which causes death of 50% of the cells. So so we need to make a curve, like we have to use different concentration. We have to use 1 millimolar, 5 millimolar, 10 millimolar, like that concentration of drugs, and we have to finally agree upon a concentration which does not cause much cell death and also does not also shows some effect. Like it should not be like it is totally toxic free, like totally it is showing no toxicity, but also in that concentration it is not showing any kind of effect. So we have to optimize that concentration depending upon the LC50 value and IC50 value of the drugs. Also, we have to standardize at what time we need to incubate, because different drugs require different time for its mechanism of action. So once all those things are standardized, then only we have to compare it with the control and check which drug is behaving the best. So before that, we already have to do the standardization of that which concentration we need to use and what time duration we need to incubate the cells with. So when we are having control X, Y, Z, different, this thing, different drugs, then so if we are testing it for Alzheimer's, for example, if we are testing the drug for Alzheimer's, so Alzheimer's, what happens is all these hippocampal neurons, they tend to die, and because of their death, there is very less hippocampal neurons and there are less synaptic junctions that are present. So this is one hippocampal neuron, and suppose this is another hippocampal neuron. So if the cells are dying, then this junction will become less, because there will be less number of neurons to form that synapse. So if the drug is working, then the synapses should be higher in those those drug treated cells. But if this drug is not working, then we have to work with different concentration of drug and see whether the effect is happening. Also, in order to be more relevant, it is also suggested that these hippocampal cells, they need to be treated with chemicals that can show Alzheimer's like effect. So just isolating hippocampal neurons and plating it and then checking the effect of the drug, it's not a very good idea, because those hippocampal neurons that we are isolating it from the fetus, those fetus, it might be a healthy fetus, and they might not show similar characteristic to what happens in diseased condition or Alzheimer's patient. So ideally it needs to be, so we need to have two controls basically. So one control will have nothing, no drug and no Alzheimer's induction also, and another control need to have Alzheimer's induction cells, like the hippocampal neuron should be induced with chemicals to show Alzheimer's like effect. So in that condition, it will be more more physiologically relevant, and the results will be more believable, because the cells which are behaving like Alzheimer's patient hippocampal cells, those there only we have to give drug treatment and check the effect. So for checking that, we can use a technique which is fluorescence labeling. So there are proteins which are synapsins or synaptophysin. So these proteins, they are present in the synaptic junction, and maybe we can label it. So once we are treating the cells with different drug, and then we have then we can use immunohistochemistry, where antibody and fluorescently labeled antibody can bind to your synapsin protein, and where there is more synapses, it will fluoresce more. Where there is less synapses, it will fluoresce less. So in experiments such as this, it is not mandatory to do it in live cells. We can fix the cells and then check the number of synapses that is formed. We are not actually looking into the molecular pathway or the live cells method. We are just checking whether the drug is working by checking the number of synapses that is expressed. So the number of synapses can be monitored using fluorescently labeled proteins against specific proteins which are present in the synapses. Next, we want to, after we have done the experiment, statistical analysis is very important, and here it is what makes a huge difference, because if it is not relevant, if the p-value, so p-value is determines the statistical significance, like how significant your data is with respect to your control. So if the p-value is not less than 0.05 and it is insignificant data, then there is, I mean, so there is no point of going ahead with the drug, because the drug is not working. So the statistical analysis needs to be performed in biological triplicate experiment, not in technical triplicate. So basically, when we are doing control and drug treated experiment, this needs to be repeated 3 times and 3 different days using 3 different passage number of cells, and we have to be sure that every time we are getting the same result, and the difference should be statistically significant. So the statistical significance will allow us to move further into, whether the drugs, we have to use it further in our in our cell culture methods, or whether the drug can go for the animal trials or not. So next, there are certain drawbacks of some hippocampal neuron drug testing. For example, one is the random culture, so when we are, so these organs are not very huge organs. I mean, hippocampus, if you are seeing, if you're some small organs, and so when we are actually taking the cells, or when we are mechanically dissociating the cells, we are taking the entire organ, dissociating the cells, and then growing it in our required plates. But so that does not eliminate the possibility that the cells are from different regions. So this region cells are there, this region cells are there, so that the mixture is not very homogeneous. So we have a heterogeneous population, and so it is not a particular area, so different cells might behave differently. So the results might not be very believable, very trustworthy, but the average, so we need to do the average of all the behavior of the cells from all the parts. So that is one drawback that happens mostly. Also, because it's a 2D culture, if we are culturing the cells in 2-dimensional, then we are not mimicking the physiological condition, because in the physiological condition inside brain, it is surrounded with so many other tissues, and it is not growing as a 2D plate, but it is surrounded with a lot of other cells, and there is crosstalk between all the cells, and not only in the XY direction but also in the Z direction. So that also affects the behavior of the cells to a greater extent. So 2D culture is also one of the drawback, and the solution is to grow the cells in 3D and check again whether the drug is showing similar effect or not. Another drawback is F18, or the fetus derived, like the fetus of 18 days. The hippocampal neurons derived from fetus of 18 days, but these neurons that are isolated from fetus, might not be relevant to adult neurons. So it might vary. The results might vary. So again, so human isolation and testing from neuron from adults again is a challenging task. I mean, because we need to get some samples. Like if you are directly want to test it in human samples, it is very difficult to get human samples for any kind of scientific experiment. There is a lot of ethical issues that comes, and so there is a lot of ethical clearances that needs to be done for testing this hypothesis. But anyway, these are some of the drawbacks, and definitely these drawbacks need to be overcome in future, and there will be some other methods by which this could be overcome. So let's see. And so the hippocampal growth neurons, the medium that is required is a neurobasal medium, or B27, because it is supplemented with glutamax. So glutamax is a dipeptide of glutamine and alanine, and hippocampal neurons requires a good amount of glutamine and alanine for their growth. Also, it is supplemented with antibiotic and antimycotic. So, antibiotic and antimycotic, it is generally known as anti-anti. And anti-anti is required to avoid contamination in your cells, and because they're primary cells, and we are isolating it from the animals, so it has to be in a very, very sterile condition, because anyway inside the body of the animal it is very sterile, but when people are dissecting it in outside, although they're dissecting it in hood, not outside, but still they have to maintain a lot of like lot of procedures, where they have to wear gloves and mask and the lab coat, proper shower cap, in order to do the dissection and then isolate cells without any contamination. So it needs to be so anyway the media needs to be supplemented with antibiotic and antimycotic, so that any kind of like small contamination, if it is there, it could be tackled with it. Also, after that, we need to poly D lysine coating, it needs to be done, so that sorry, polylysine coating needs to be done, so that it's like the hippocampal cells are grown in plates containing polylysine, so that it can grow properly. And then media composition and cell culture conditions must be optimized in such a way that if there is a mixture of hippocampal and glial cells, so when we are isolating hippocampal cells, there might be some glial cells that can come into the mixture, because that organ is like surrounded with glial cells, and they have some glial cells. It's not possible that you can remove the possibility of having glial cells or other accessory neuronal cells. It is very much possible that we have contaminating cells, but the problem with contaminating cells is glial cells are dividing cells, and they can divide. So when we are isolating hippocampal cell and glial cells together, if there is any contamination, so then the glial cells might divide and overpopulate the non-dividing hippocampal cells. So we have to optimize the media composition in such a way that it will supplement the growth of only hippocampus and not the glial cells, because we don't want that the glial cells should divide and should replicate very fast as compared to the hippocampus. We don't want that. So we want so we need to get a media composition, and also the culturing conditions should be optimized in a way that we get only hippocampal neurons, and they can grow irrespective of like small amount of glial cells. So that is all for today's lecture, and so there is any questions? Any questions, anybody? Also, I have uploaded all the video links, YouTube links till week 3, week 4, and week 5. I'll upload this week, and also the slides also I have shared. So you can go through all the slides and the recorded lectures of the past 4 weeks recorded lecture and the slides. So yeah, you can go through them. I have already updated it. Week 4 and week 5 recorded lecture I will upload today. Today, not this week, maybe. Yeah. So any questions, anybody?
The defined media, how expensive are the defined media?
How?
How expensive? Cost?
Expensive. See, defined media means any media that is not having serum. So basically, serum is anyway we are getting it as a separate separate box. Like fetal bovine serum, we get it as a separate separate bottle. So any DMEM media that I was talking about, that is actually defined media only, that does not have any serum in it. So it is around how much I will say. So there are two categories. One is like powdered media, like that media you have to add autoclave milk water and filter it, and another is liquid media where we don't have to add anything, and you just need to filter and use it. So the powdered media is around some 6,000, 7,000 rupees for 10 packets. Like 10 packets in the sense one packet can make 1 liter of the media. So 10 liters of media is around 6 to 7,000 for defined media. And the serum is actually expensive because the fetal bovine serum, they are isolating it from calf. So one bottle of serum comes around, I think, 26 or 30,000 something. So one bottle in the sense 500 ml. Just 500 ml is around 30,000. So serum is expensive. The normal media, like without the serum, is okay-ish. Okay. Okay, so let us go to the question answers. Okay, so you can reply in the chat. Yes, so defined media is something which does not have serum. So presence of which will disqualify a culture medium as defined is serum. So that's right, option C.
Ma'am?
Yes.
But we are using fetal bovine medium, right? Say, like serum, right? It has serum or not? Then
Fetal bovine serum is a separate bottle. I mean, when we are making media for the cells, we can add fetal bovine serum. Like basically, one bottle. Suppose if we have one bottle of DMEM, so one packet. Suppose one packet is having 1 liter, 1 liter amount of media. So if we are putting 1 liter water, and then we add 1 liter water over here, so 1,000 ml water, and so we can use it as it is without adding fetal bovine serum. So if we are adding fetal bovine serum, then the fetal bovine serum is coming in separate bottle. So this fetal bovine serum, then from this 1,000 ml of the DMEM that we are preparing, we have to actually take out 100 ml of DMEM and add 100 ml of FBS. So add 100 ml of FBS to 900 ml of 900 ml of DMEM. So that media will become now media that contains FBS. So otherwise, the media, like the powdered packet or any other media composition that we get, that does not have serum in it inbuilt. Serum is always like serum. We can add it depending upon our requirement, if we need it, and how much percentage we need it, that also it can be added. So it is not like already added kind of thing in the media. It is separate. So you can add it separate. You can add it if you require it. If you don't require it, you can just add water to that powder and just use it. So in our lab, we have we use 10% FBS all the time. So in this 1 liter packet of the media that we have as 1 liter packet, to that we add 1000 ml of water. Then we properly mix the powder so that the powder is totally dissolved in the water. In that powder, we then remove 100 ml. We remove 100 ml of that water which contains the media, like the 100 ml of the DMEM media without serum, and that 100 ml we can use it for if we are doing if we need opti-MEM media for transfection. So some kind of transfection reagents like lipofectamine and all, they require transfection to be done in without serum media, because those are proteins, and those proteins can transfection reagent, and those things are some interaction they cause, as a result of which transfection is not proper. But the transfection reagent that we use is jet prime, and jet prime works fine with serum. So we don't need to use separate media which does not have serum. So we generally have 1000 in 1000 ml of 1000 ml of media, 100 ml media we take out and keep, and we add 100 ml FBS to it. So 900 ml of DMEM, and 900 ml DMEM and 100 ml FBS. So that we call it as complete media, or CDMEM. CDMEM means complete media. So if we are adding 900 ml DMEM and we add 100 ml FBS, 100 ml FBS. So that is, so this media and all, we have to prepare it. We have to prepare it in filtering apparatus. As I discussed in one of the class, that there is this filtering apparatus where we have to filter all this media. So that time only we have to add the serum in this media. So this is known as CDMEM or complete DMEM. So this is what we use generally for cell culture, because this works fine with our cells. I mean, 10% FBS is fine. This is 10% FBS. So this works fine. Also, as I said, FBS needs to be, first inactivated and then used. So, yeah. The next question then. Right. So Alzheimer's disease affects the hippocampus part of the brain. So this is the correct option. Next question. Yes, so papain is used for enzymatic dissociation, so that's correct. Next question. So Revant, we can find viable cell concentration as well as the total cell concentration, both from hemocytometer. So hemocytometer, if we are staining it, so the cells, if we are staining it with trypan blue, the cells which take up blue color, those are dead cells. The cells which do not take up the dye, those are live cells. So you can count the number of live cells by counting the number of cells which has not taken up the dye, and also count the number of dead cells by counting the number of cells that has taken up the dye, that is blue color cells. So you can add the number of live cell plus dead cells, so you can get the total cell count. But generally, for using it in for experiments, we have to go with the viable cell count. So then it is like more relevant because you are using those many viable cells for experiment. But we can, to answer your question, we can count both viable and dead cells. Okay, so I will help you with the formula. So what is the formula for counting? So for counting the cell is number of cells is equal to number of cells per ml. So that is equal to total number of cells total number of counted cells cells into 10 to the power 4 into dilution factor. So I will write as DF, that is dilution factor, divided by the number of squares that you are taking into consideration. So we have 100 cells in 4 corner squares, right? So the total number of cells is 100. And we have number of squares that we are taking cells are 4. And so we have to multiply it with 10 to the power 4 according to the formula. And what is the dilution factor here? Basically, what is the dilution factor? So you are taking 10 microliter of cell suspension and 10 microliter of trypan blue, correct? So Samiksha, so we have 1:2. So the dilution is 1:2 because we are adding 10 microliter suspension and 10 microliter trypan blue. So the dilution factor is 2. So if the dilution factor is 2, then the answer becomes as approximately 500,000. So the correct answer is option number D. Okay. So depending upon how much you're diluting it, there your number of cells will vary. So don't consider how much amount you're loading. Consider where how much amount you have diluted and then you have loaded basically. So even if you have loaded 10 microliter, it is 1:2 dilution only because already the cell suspension were initially diluted with trypan blue, so it has half the amount of cell as compared to the original suspension. So the dilution factor is 2 itself. So, whatever amount of trypan blue you are adding and to the cell suspension, depending upon that, the dilution factor is changing, and so that affects the dilution factor and not how much you are loading. So, next question we'll see. So this is option.
Madam, dilution factor can you repeat, madam? 10 microliters plus 10 microliter. Is it 1:1 or 1:2, madam?
It is 1:2. I mean, if you are saying the how much dilute, so 1:1 not, because you are adding 1:1 amount of cell suspension is to trypan blue. But if you see that total volume is how much? 20 microliter, right? So you are adding 10 microliter of cell suspension and 10 microliter of trypan blue. So in 20 microliter, we only have 10 microliter of cells, 10 microliter of cell suspension. So the dilution is 1:2.
Okay.
Okay. So, but the dilution factor is 2. Dilution factor has to be 2, not 1:2. Dilution is 1:2. Dilution factor is 2.
1:1 means, madam?
1:1 means if you are adding, suppose 1:1 will be 1:1 dilution is like that is not dilution. Basically, you will dilution means you have to either add some amount of water. So if you are suppose taking 100 microliter of your sample, okay, and putting it in 2 dishes, like suppose 2 dishes where you're adding 50 microliter of the sample like cells, then it becomes 1:1 splitting. So that is 1:1 splitting means out of 100 microliter of cells. So in this appendot, you have 100 microliter of cells and you are adding 50 microliter. That is 1:1 splitting because you are adding equal amount of cells in each of the dish. But dilution, where you are adding 10 microliter of suspension and 10 microliter of trypan blue, that is 1:2 dilution because here the total volume is 20 microliter. And 1:1 dilution, 1:1 dilution, how will you calculate? There is if you are adding, so 10 microliter of cell suspension and again 10 microliter of that is 1:2 dilution only. 1:1 dilution is not, I don't know whether, I don't know. I mean, 1:1 cell dilution is not, I've never thought it about that, but no, I don't know. Maybe I have to check.
It's okay, madam.
Yeah. Okay, Revan. So again, I will explain. So basically, if you are having 10 microliter of your cells suspension, and you're adding 10 microliter. So what is dilution and what is splitting? I will explain the difference between it. So basically, when you have cell suspension, suppose you have 100 microliter of your cell suspension over here. Now in this 100 microliter, you are adding 100 microliter trypan blue. So if you're adding 100 microliter trypan blue, or if you're adding any kind of thing, if you're adding water also, if you're adding trypan blue or if you're adding water, so now your total volume becomes how much? Your total volume becomes 100 plus 100, 200. So total volume is now 200. Total volume is 200. So if in this 200 microliter volume, how much microliter is your cells? So 100 microliter cells is available in 200 microliter volume. So dilution is 1:2 because in 100 microliter of cells, in 200 microliter of total volume, we have 100 microliter of cells. So that is the ratio, 1:2, not 1:200, 1:2. Always dilution has to be calculated with the total volume, how much total volume you are adding. For example, if you are adding 100 microliter of cell sample and 500 microliter of your water or trypan blue, so now the total volume is 500 microliter of your water or trypan blue. Now the total volume is how much? 600 microliter. So out of 600 microliter, you have 100 microliter of your cell sample. Okay, 100 microliter of your cell sample. So here the ratio is 1:6. So the dilution is 1:6. So that is what is dilution. Okay. Now, when we have dilution factor, that factor is opposite to the dilution. So 1:6 means 6 is the dilution factor. So, when we split the cells, suppose we are splitting cells, same 100 microliter of cells, if we are splitting it into 50 microliter and 50 microliter, okay, if you are splitting it into 50 microliter and 50 microliter, that means there is equal number of splitting, because in 100 microliter of cells, if there was about 5 lakh cell, then out of that we have put 50 microliter, 50 microliter. So we have put 2 lakh 50,000, 2 lakh 50,000 cell. So that is equal splitting. So here we say it as 1:1 splitting. 1:1 splitting, because equal number of cells we are seeding it in the dishes. This we are not diluting, this we are splitting it. Diluting it is when we are using like the same stock and adding extra water or extra buffer. That is what is diluting. Here we are not diluting. Here we are just seeding the cells in such a way that there is equal number of seeding density in both the dishes. So dilution means when you have to consider the total volume of how much after adding the water, how much. So basically when we are using the formula V1S1, I think don't know. So you have been, you might be knowing this formula, V1S1 equals to V2S2. So always the dilution is considered with the total volume, not the initial volume. Like we are not considering 10:10 as 1:1 split, 1:1 dilution, because the total volume here is 20 microliter. The total volume after adding trypan blue becomes 20 microliter. So the dilution is also considered after adding trypan blue, how much volume is there, and in that volume how much of your cell suspension is there. So in 20 microliter, if 10 microliter cell suspension, that means 1:2 splitting. If in 40 microliter, 10 microliter cell suspension means 1:5 splitting. Okay, 40 microliter if there is trypan blue and only 10 microliter of cell suspension, that means total volume is 50 microliter. In 50 microliter, you have 10 microliter cell suspension. That means 1:5 splitting, because 40 plus 10 is equals to 50, and in total volume 50, we have 10 microliter of cells. So that is 1:5 splitting, 1:5 dilution, sorry. Okay.
Madam, can you explain the seeding density according to 96 well plate to 6 well plate to 48 well plates, madam? If any
So,
standard guidelines?
Standard guidelines is like, so if you are seeding it with in a very small plates, 96 well plate or 48 well plate, those wells are really small. So what we prefer to do is we don't and also very practical one problem that people can face is, for example, if you are seeding it in dishes, 35 mm dishes, you can easily circle the cells and properly mix the the inoculum of cells with the rest of the media, so that the cells will be properly distributed. But for 96 well plate and 48 well plate, you cannot swirl it. You cannot swirl the plates, and it will, because the area is very small. So what we do is, suppose we have 135 dish and we have to seed equally in 96 well plate. Also, we have to keep in mind, because the area is very small, so the cells might get overconfluent very easily. So we take very less amount of cells. So for example, 135 dish, which is around not very confluent, around 50% confluency 35 dish, we will maybe dilute it to again half of that. So 50% confluency 35 dish, suppose we are getting the cell pellet as some pellet we are getting, and in that pellet we will add 200 microliter of media. Out of this 200 microliter of media, we will just discard 100 microliter media, and to the rest of the 100 microliter media we can add 1 ml of so out of 200 microliter of media, suppose you are discarding 100 microliter, so now, although you are discarding 100 microliter of media, the rest 100 microliter that is there, that is actually 1:2 only, because you have we had earlier added 200 microliter of media, and from there 100 microliter we are discarding. So we have just now whatever cells were present initially in the pellet, half the cells I have discarded, and only half the cells are present. So those half cells, then we put it in 1 ml Eppendorf, and put 1 ml of 1 ml of media, again fresh media, to the 100 microliter cells. We again put 1 ml fresh media, and this 1 ml fresh media and 100 microliter cell, we properly mix it in the tube itself, pipette it once or twice, and then mix it by just rotating it up and down. And once we have done it, and then we can plate it in 96 well plate. So per 96 well plate, one well, we can either plate at 100 microliter, or we can plate 200 microliter, depending upon what your lab follows. But our lab, generally we plate around 200 microliter or 100 microliter. So yes, so basically we have to, when we are plating it in 96 well plate or 48 well plate, we have to always we have to mix the cells well in the Eppendorf tube or in the Falcon tubes properly before aliquating it, because when you are aliquating it, after aliquating it, you cannot mix it in the 96 well plate. But generally bigger dishes, 35 dishes or 60 or T25 flasks, those are big. The surface area is big, so once we are this 100 microliter media, we don't need to again resuspend it in 1 ml of media and then put it, because this 100 microliter media, you can put it directly in that flask. So that flask will already have the standard amount of media. So for T25 flask, we need to grow the cells in 3 ml of media. For 60 also 3 ml of media. For 35 1.5 ml media. So like that is like in that media itself, if you are putting that inoculum and just mixing it, it will be fine. But for 96 well plate and 48 well plate, we have to instead of mixing it there, we have to put it better, like we have to mix it in the Eppendorf tubes or Falcon tubes only, so that it is not getting spread, so that it will get spread properly. Yes, Revant, then we will, I will upload questions like this again in next class. So I hope the splitting and the dilution is clear now. Okay, so let's go to next question now then.
Memory.
Yeah, correct. Okay, everyone has given the correct answer. Let's go to the next question. So Alzheimer's disease leads to loss of memory. This question, I think somebody had already discussed earlier, I guess, some earlier class.
Within the class.
Yeah, you can write it in the chat box so that everybody can, I can see. Correct. So in vitro means within the glass. So within the glass, and like the word itself, the Latin word phrase itself is like within petri. So sorry, within test tube or glass. So it's within the glass. But actually, in vitro nowadays we can do it in plastic also. The plastic wares are also fine for in vitro cultures. So the term specifically means that, yes. So on silicon means, so these are some lab on chip methods, some silicon. So if some kind of like silica coating is present or silicon material is present, then you can grow it. But I don't have much idea on like what silicon, what kind of cells or what kind of growth happens there. So. Yeah. So now next, go to next question. Ma'am, all in vitro cultures will be in glass only. No, actually, so initially when this in vitro term was introduced, that time, so scientists used to use test tubes and glass dishes. But over the period of time now that we are using, we don't have access, not access also, but glass is like not very feasible. It might break, it might cause problems. So nowadays in vitro cultures are also done in plastic as well. So it is not like only in glass, just the term, because when it was first coined, the term actually meant it was they were doing it in glass dishes or glass test tubes. But nowadays people are, we also in fact do it in plastic wares. So in order to reduce cost and also to avoid the chances of glass breaking and hurting. So yeah. Yeah. So now next question again. Right. So in vivo means within the living. So that's the correct option. Driven by in situ. Okay, so the correct answer is C, that is locally. In situ is not, option A is not correct because within the living is within the living means in vivo, and in situ is generally used as a like it's a very general term, not just for biological people. In vitro and in vivo as is like mostly biological people, but in situ is a more general term. We also use it in other aspects. So in situ means in localized condition or in like nearby places, not global global present, like if it is present in local nearby areas, those are in situ. So global area, it should not be present. So option number C is correct for in situ. The last question for today. Okay, so most of you have given the correct answer. It's not option A, it's not glutamine. It's not glutamine. Glutamax is a dipeptide and it consists of both glutamine and alanine. So the option number C is correct. Okay, so that's all for today. If you have any questions, then let me know.
Ma'am, I have a question.
Yeah.
Ma'am, if we use to hematite and for a total cell count, if
Uh,
we add the suspension, the red boundary, they also spill out of the red boundary. If we are not counted, the cells less count comes, then the error may.
sorry, can you again tell me the question? What the cells which are in the red boundary? We have what you asked, whether we have to count it or not.
Out of red boundary will not count.
So basically, cells. So let me share it again. The red boundary here, I have not put anything. I just because we didn't need to know the cells where they had already given the number of cells here. But if you had to actually count the cells, then ideally it is uh, ideally it is like we should count cells which are on the top and on the right side, and not the cells which are in the below or on bottom side or on the left side. So basically these cells which are present over here, cells which are present over here, those cells we are not supposed to count. Okay, but cells which are present over here and cells which are present over here, those cells we can count. That is true for each of the grid, like the smaller grids also. If the cells are towards the right and towards the top, that those cells we can count.
Now we count inside the red boundary.
So here, here there is no red boundary as such. So here you, so which is the red boundary? I don't know. Where red boundary?
Is shown in the figure, not
Oh
hematite
no
and
no, yeah
graphite.
yeah, that figure I have not gone through that figure. So because here we did not. Yeah, indicated in the red boundary of this image. Yes, here actually the figure I have not actually downloaded because they have already given the number of cells that they have got. So I think I will just go see the question again. Basically, it would mean the cells which are also included in that red boundary. Those cells, including those cells, we are getting 100 cells. So yeah, so but the main idea behind it is that we should not count cells which are in the bottom and towards the left, and we can count the cells which are towards the top and towards the right. So here this part you totally ignore it because I have not put the image here. So there is no point, right? Because we already know how many number of cells are there. If you had to count it, then this would have been required, because you would require that where the cells are exactly in the boundary, or whether it is towards the left, right, wherever. So but here you are already getting how many number of cells are there. So here this part you can ignore, because I have not put the picture of the image over here. Any other questions? So Shweta, what is the role of Glutamax? So Glutamax, that has a more amount of glutamine and alanine. So this is required for, I think, these neuronal cells, the hippocampal neurons, they need this amino acids for their better growth. So basically, it will be required for their, I think, some cellular functions, as a result of which we need it. Exactly what is the function is they have like more amount of those amino acids for their like for their cell proliferation and cell growth. So for that reason only we need it. And so reference books, I generally, there is these handbooks available in Thermo Fisher website, also in Eppendorf websites. So those handbooks work fine. Otherwise, I also have one handbook, I don't know from which website, either Sigma or Evolution, sorry, say Sigma or Thermo Fisher. Maybe I will check once and I'll tell you. I had one one handbook. I have to check. I'll tell you the next class. Otherwise, this Eppendorf, Sigma, the other, what is the Thermo Fisher, they have good handbooks available. So you can download it and then you can go through it. I mean, it will be better if you have some kind of practical exposure, because it might become boring without like actually knowing it, what you are going, like what you are studying. So it is better, I think, if you have some practical exposure, then you will find interest in studying those, books. Everything has a particular logic, but without, I mean, practical experience, it might be a little boring, I think. But yeah.
Madam, from where can we get the defined medium?
From where can we get
From
it?
where we can? Yeah,
So
exactly.
company, Jipco company supplies it. Jipco, you can look. We use Jipco media only in our lab. So Trishla has asked, Scientist X has 500,000 cells in 500 microliter as the cell suspension. X decides to plate on 15 mm round cover slips at 500 cells per mm square. What volume should X add to how to calculate for round cover slips? So for round cover slips, we will calculate the surface area, basically pi r square, I think. So here they have given the radius. This has to plate on 15 mm round cover slip. So 15 mm round cover slip, 15 mm is the diameter. So diameter is 15 mm, so radius is 7.5 mm. So you can, I mean, calculate it as 7.5 pi r square. So we have to, what volume should X add from the cell suspension per cover slip to achieve the desired plating density. So let us do once. 1 ml is 1000 microliter, and if they have 500,000 cells in 500 microliter. So X decides to plate cells on this at 500 cells So in 1 millimeter square, we need to split 500 cells. Okay. So 500 cells in 1 microliter we have how many cells? In 1 microliter we have 500,000 divided by 500 cells. So that is 1,000 cells. So 1 microliter has 1,000 cells. So we need to have so in 1 microliter has 1,000 cells. In 1 ml it will be 1 cell. So we need to put 500 cells per mm square. So 1 mm into 1 mm, we need to have 500 cells. So in total surface area, ground covers at 500 cells per mm square. What volume should X add from the surface tension to achieve the desired breathing density? So 1 mm square is 100 cells and and 7 divided by 4 is 96. 176.7, so that is the area. In this area, we have to have 1 mm into 100 cells. So we need to put 500 cells in. I think the third option is correct here, 88 microliter. What is the option? Okay. So I will. What is the correct answer? I will share my screen. So let me. So is the answer 88 microliter, Trishla? Yeah, so I will share my screen. So let me. Okay, so basically they are saying that in 500 microliter you have 500,000 cells. Okay, so 500 microliter has 500,000 cells. So 500,000 cells. So 1 microliter has how many cells? 1 microliter has 1000 cells. Okay. 1 microliter has then 1000 cells. 1000 cells. So now we have to plate the cells in round cover slip in such a way that 1 millimeter square, so 1 millimeter into 1 millimeter square, 1 millimeter square should have around 500 cells, right? So 1 millimeter square should have around 500 cells. Now they have given that the cover slip is round. So round cover slip's area is how much? Area is surface area is pi r square. Okay. So round cover slip surface area is pi r square. So they have given as 15 mm round cover slip. So 15 mm means diameter is 15 mm. So 15 mm is the diameter of the cover slip. Okay. So we have 15 mm as D. So the pi r square will be 22 by 7 divided by multiplied by 15 divided by 2 whole square. Okay, so this comes around the area comes around 176.7. This comes around 176.7. 176.7 mm square. So this area is 167.7 mm square. So in this mm, how much cells we need? That means, so if in 1 mm we need 500 cells, that means in the whole cover slip we need 176.7 into 500 cells, right? In the whole cover slip we need that much amount of cells, 166.7 into 500 cells. So total we have to plate equals to some 88,350 cells. 88,350 cells. Okay. So now if in this whole cover slip, we have to plate around 88,350 cells. So how much volume we have to add so that whole cover slip gets, filled up in such a way that 1 millimeter square is 500 cell. So for 1 microliter, it is 1000 cell. So for 88,350 cells, how many microliter we have to add? So 88,350, again, I will put it over here. So 88,350 divided by 1000. So that much amount of that microliter of cells, that much microliter you have to add so that you get divided by 1000. So that is approximately 88 microliter. Okay, understood. Okay, Manasa is asking, ma'am, does glutamine and glutamate has same chemical formula? No. Glutamine is just glutamine. Glutamine is just the amino acid glutamine that has that extra amino group. But glutamax consists of glutamine plus alanine, so it has a mixture of two different amino acids. So the composition is not same because glutamax consists of glutamine and alanine. Sorry, and glutamine is just glutamine. Okay, so next class, can you explain about cell storage? I have discussed about cryopreservation in one of the earlier classes. I have already uploaded the recorded versions, recorded lectures. Only last week's lecture and today's lecture I will upload it this week itself. So you can go through the lectures in my YouTube channel, and yes, and the slides also you can access it. So yeah, so that is it. So okay, then any more other questions? Otherwise, next class again we will start from other questions if you have. So okay, then we will say, then we will be, yeah, we'll just say bye bye and we'll again meet next week. So again next week Monday 6 p.m. Okay, thank you everyone. Thank you for attending the class. Bye.